So I want to thank each of our panel members again for their ability to come here today and to speak. We wanted to make sure that we had ample time to open this up for Q&A. So any question, obviously, as you can tell, is fair game. Depending on the answers that you get, we'll just soon tell. But just to kind of kick off the conversation, and I guess we'll have to pass a mic down and share it there. I want to start with a couple of leading questions. This one probably is most relevant for Julie. We've heard this morning, obviously, there are some very unique physical phenomenon that are associated with microgravity that can be advantageous in biological research. And clearly, I think there is a lot of evidence that can be pointed towards terrestrial biomedical outcomes and other opportunities. I'm curious, though, on the International Space Station today, what are the types of research models or research facilities are available that we can take advantage of?
Yeah, so it's great you asked that question, Dwayne, because we were joking at dinner last night. We were trying to count the kinds of organisms that have been on the space station so far, and we got well over 10. So there are, of course, the classic facilities that biomedical researchers have asked for over the years. The ability to fly a mouse model, will eventually upgrade that to also being able to be used for a rat model. But we're starting with mice just to get a higher sample size for investigations that can use marine models. We have Drosophila capabilities that are starting this year, and one thing I learned along the way with that is that over 18% of the research funded by NIH uses Drosophila as a model, so that's a huge opportunity to understand genetic functioning and use microgravity to take gravity out of the equation and see what a gene does in response to that. Of course, the C. elegans, you saw those models. There are a number of different plant facilities. We have unique facilities that have primarily been used for education that can culture spiders and butterflies and a variety of insects beyond Drosophila. And of course, then there's the good old humanist subject, which offers significant opportunities and has a really nice, rich past database of research that's been done. NASA's research has always been focused on how to be go for Mars, or how to be go for the moon, or go for an asteroid even. But in what they leave in their wake is they treat the human as almost another system in the engineering structure of a space mission. What they leave in their wake is all kinds of information about what's going wrong and why things are working that nobody at NASA really follows up on, because once they've solved the problem, they're heading on out. And so there's this really rich literature that you may not always see, because if you're not focused necessarily, say your interest is bone loss, if you're not focused on you might just skip over a space article saying, well, that's not relevant to me, when in actuality, there could be a nugget in there that is incredibly relevant to a set of research that's going on today.
So with respect to astronauts as research subjects, my assumption is sample size is relatively low, and are there ways to address that?
Yeah, so generally, our studies that have been done with astronauts as subjects are low power. Between 12 and 25 subjects have been typical for most investigations. So they're definitely not like a clinical study where you can enroll 1,000 people. That said, they're a very homogeneous group. So if you're doing an osteoporosis study on astronauts, you've got a set of people who are fit, who have no other medical issues, and the only thing that's going to happen to them is they're going to be in space and have a reduction in the muscle contractions and things on their bone, and that's it. So it's a very clean kind of a subject group. When they come home, they're also fit, they're committed to recovering quickly, and so even though it's a small sample size, it's also a unique group, and that's valuable as well.
Cliff, you touched on this a little bit in your earlier presentation, but I'm curious if you can maybe sort of relate evolutionary biology to the microgravity environment.
Yeah, so evolutionary, all biology can only be understood all of biology has to be understood in the context of evolution, and many of the functions that we're looking at, the really very fundamental cellular functions that we're looking at, are conserved. Having said that, when you get into the area of embryonic stem cells and you get into the area of cancer biology, you're dealing with highly variable post-translational modifications and DNA methylations that change the course from a strict Mendelian biology into more of an epigenetic-based biology. The microgravity environment takes out one extraordinarily important adaptive function of all cells, which is staying upright. So we devote an enormous amount of our energy to keeping our cellular integrity intact and standing up. Once you take that out, the energetic equation of the cell changes dramatically. And so you can start to study issues in, for example, embryonic stem cells, not only because the colloid function of the cytoskeleton of the cell changes, as Joel discussed, but also the cell growth characteristics change. And as opposed to, for example, the embryonic stem cell world of getting tiny little embryoid bodies, you can get large amounts of cell mass in growth, and then, as a consequence, large amounts of cell product. So all of a sudden, you're dealing in a very different space. When you're not doing 1 or 2 or 3 or 10 cells, you can be doing orders of magnitude more and orders of magnitude more cell product. So the whole issue of evolutionary biology is important. And then the last thing, of course, and this is way out of my area, but has been extremely well studied over time and will continue to be, is the effects of ionizing radiation. Because ionizing radiation, obviously, even in the shielded environment of the ISS, is a very different fish than it is even for high altitude jet flight.
So I can only imagine that, and this question is for Gwen and Robert actually, that both of you are used to and have quite a bit of experience in going to the benchtop and conducting your research in a traditional laboratory environment. I'm wondering if you can comment on what did you have to do differently, or what are the obvious differences that you've had to encounter in order to still have success with your research objectives, but to sort of tailor it to meet the ISS laboratory environment?
So just to echo a few things earlier, it is nice from my research, where I deal normally with a very, very heterogeneous population, have a very narrow set of very homogeneous. They're healthy before, there's a significant change during, and then they're again normal, hopefully afterwards. But from one of the changes for me moving forward with just the surprise that I could do this research, the opportunity to do this research was really just such a great opportunity, and really stumbling upon, just like Julie mentioned, maybe this space paper I'd never really looked at. I'd been mainly focused on a bunch of other different areas, and then stumbled upon a paper on space-based research and realized that there are changes that would be very relevant to the research that I'm doing currently and would help define some of the parameters moving forward.
Much of what we had to work on is we work with droplets, with solutions, and those all float in microgravity. And so trying to make our experiments work such that these solutions would stay in the places where we needed them to, so the experiment would work. And so we worked with a team of engineers to make sure we had a system where we could do the experiments we needed to.
Okay. You mentioned, Robert, that there was access to historical research that had been done. One of the criticisms that we sometimes hear is that there's either not a wealth of peer-reviewed and published literature, or they just haven't quite gotten into that academic environment yet. Julie, are there other resources where we can tap into in order to better understand what may have been contemplated or questions that have been asked in the past that can help us in generating research objectives in the future?
Yeah, so on NASA.gov, we keep a comprehensive database that you can sort by discipline and subdiscipline to go and sort of quickly catch up on what research has been done in those areas and browse all of the publications that have come out from the space station. It also cross-references back to some of the old things, the things from Mercury and Gemini and so forth. If there's a trend for those, there'll be a related papers section on that website, so you can actually dig all the way back in through the related shuttle research, and really, so it's a way to catch up on a discipline pretty quickly.
So Gwen, your research is actually taking place right now.
Yes.
I'm really curious as to what exactly is going on while on the International Space Station, but probably I'm more interested in what do you expect to get back and what do you do next?
Right. So once our experiments reached the International Space Station, the next day they were activated by the astronauts on board. And right now they're just hanging out in a 4°C locker. And they'll remain there until they'll come back on SpaceX 4. When they come back down on SpaceX 4, we hope that there'll be crystals of these proteins that have been really, really challenging to crystallize on Earth. And so if we have crystals of any of these proteins, we'll do X-ray diffraction on these proteins and hopefully get a crystal structure that we've never been able to get before.
So in your current protocol, the imaging will take place after the exposure and the samples, if you will, are brought back.
Exactly, yes.
My understanding, Julie, is that there will be diffraction capability on orbit in the near future?
Yeah, there is a diffraction capability being developed right now that will be available soon.
So Cliff, I'm curious, you talk a lot this morning about genomic studies, and obviously those are most times huge population studies, yet we're contemplating doing this research in an environment where we either have a sample size of 3 or not a robust amount of samples. How do you address that, and why microgravity with respect to omics research?
Oh, that's a really great question. Thanks for the softball. And my last slides, which were the really good slides, didn't show. So I'll show you the end. I was wondering why I kept talking, and people were looking at me strangely because I was talking about a slide that I saw, but nobody else. Anyway, yes, this is really a very, very interesting and important question, and it gets to the heart of what is called omics research, which probably some people in this room are involved in. Typically, what's done in omic research, proteomic, metabolomic research, is that there's an aggregate of population. You try to make it as homozygous as possible. You try to look at middle-aged people of Cuban ancestry who have lived in the U.S. for 20 years or something. Pick a population, try to get it as narrow as you possibly can, but you never, because we are all a result of post-translational modifications, why we all look so different, even though we have the same genetic complement, is that it becomes more and more critical to start looking at each individual as what I refer to an N of 1 experiment, that each individual has his or her own genetic complement that he or she has come to in a unique way, and trying to aggregate those populations you start to lose the differentiation and the subtle changes that might turn out to be very important. So as a consequence, we are developing right now with colleagues in information sciences as well as colleagues in digital signal processing and computer science, a new analytic methodology for doing genetics, which with each person or genetics and omics, with each person functioning as his or her own control. Why this is important, it's important, and we will test it in space because, as you said, that's the world you have to live in. But it is my belief that this is critical in understanding the differential activity of chronic illness in normal populations. And those of us who are clinicians know that we call a lot of people type 2 diabetes, but they're phenotypically very, very, very different. And we've got to get away from this notion of measuring blood sugar alone as an indicator of the activity of diabetes, because we really haven't made much progress in scaling. And the stimulus of the necessity for doing that type of experiment in orbit has been driving us to create this new mathematical model, which, if we're correct, when we're correct, will have, I think, a substantial impact on human disease. In terms of cell behavior in orbit, there's just preliminary data starting to come out. There's some interesting work on Tregs. There's been some interesting work on cytoskeleton. There has been some interesting work on immune function in general. But the actual molecular biophysics of cell behavior in orbit is an area that's just beginning to be explored.
Robert, I'm curious if you can probably talk a little bit more about the research that you're doing. I understand the rationale of studying intracranial pressure in that environment, and the evidence points to the fact that you actually have an increase in ICP as a result of microgravity. But clearly your research is targeted to an impact that you'll have here back on Earth. And I'm wondering if you can just maybe talk a little bit more about how that relates. How is it that the research you're doing in space will bring a product opportunity here on the ground?
Absolutely. So I think that one of the problems initially with some of my research is it's data-driven, and clinicians at the tail end, when we have some positive results, want to know why. Why does that work? Clinicians are usually not a big fan of black box answers. So one of the problems that I have, and why I was so excited about the ISS research, is to understand some of the changes. Now, I briefly went over it in my PowerPoint presentation, but there are subtle changes that we are able to detect in that transcranial Doppler waveform that hadn't been detected before. But my difficulty now is to explain to clinicians why that's important. So using this very controlled research environment where I have measurements before, during, and after, I can look at those subtle changes and say, well, look, there's an increase in venous pressure because of this fluid redistribution, and that's causing, or at least contributing to, this morphological change. Obviously, preclinical studies will also benefit that as well, but utilizing even a small n would really take me a long way into explaining those morphological changes that I need to for this to really be a mainstream solution for non-invasive intracranial pressure detection.
So in Joel's keynote address earlier this morning, and then some of the evidence that was presented in Julie's slides, clearly in understanding how our cells work, there are a lot of issues associated with fluid transfer, fluid mechanics, as well as cell-cell interaction and how things pass across membrane barriers. So Julie, I'm curious, is there any research that has been done that looks at drug delivery or medication and whether that's beneficial or detrimental, if you will, or is there any knowledge that can be derived from that can be used towards terrestrial medicine?
Yeah, so there has been a small set of work on, say, pharmacodynamics, for example, aimed at healthcare and the crew, and it is confusing as heck. Certain drugs seem to be pretty ineffective for things that they work great on here on Earth. There are other drugs that seem to take much higher doses in order to be effective, and that's an area that I would say hasn't been well probed so far. It's one of these examples of where NASA will solve the immediate problem. If they can get pain relief with double the dose of Tylenol, that's good enough. They'll just take more Tylenol to Mars. But the question of what changes, why Tylenol's different in space, is a really interesting one, because it may tell you something about how Tylenol works that nobody knows back here on the ground.
And I appreciate your comments earlier about sort of a change with respect to how research may be conducted going on in the future. And clearly this will involve not only NASA, but CASIS in our approach. And quite frankly, the underlying issue is that it is a laboratory that has finite capabilities as well as finite real estate. And the concept or the notion of creating a one-off study where you have to invest quite a bit of time and money and resources in the development of a piece of hardware or a facility in order to satisfy one experimental notion is something that really is sort of gone the way of the past, but rather looking at research objectives that have an interest by a lot of individuals, if you will, that can be conducted, and then the results shared across an even larger population is certainly advantageous. My question is, how can any investigator influence the research that will be conducted? You mentioned a science advisory panel, but how do we interact with that science advisory panel to kind of influence the research that would be performed on ISS?
Yeah, so it's an interesting question, and one of the things that folks at NASA headquarters who are setting this up are still looking at is how to make sure that science advisory panel has all the right membership. But CASIS has been invited to name members to that panel. And so, you know, across the investigators that have relationships with CASIS, there's the opportunity to put them on the panel. And then similarly, the investigators that have existing relationships with NASA. There's also some discussion of an open call so that experts around the country who don't have relationships with anyone who's done space research or can facilitate space research can propose their approaches. And that's typically how NASA does things. So I hope that that's the decision that will be made in the future.
I'd like to open it up to the audience. Tim?
So for the recording audience, the question really was, is the microarray data that folks have showed from some past experiments, is that actually available? And the answer is, it is, in the same way that any kinds of data like that go into gene banks and into the appropriate archives as required by the journals. But it's never been integrated together into a single NASA source. And so that's one of the elements of the Gene Lab initiative, is to make sure that the metadata cross-referencing and helping you find all the related space publications looking at particular gene arrays and so forth, so that it's possible to dip out of the regular tools that you might be using in your research and grab in an understanding or data on particular genes and so forth out of all of those databases. So right now, it's a little bit of a needle in a haystack. It's there, but it's not easy. And one of the things that Gene Lab will be doing is making that easy.
Mark?
Oh, I'm gonna have nightmares about my slides not advancing.
Sure. So the way that, you know, NASA is an engineering culture and engineering organization, and so it looks at humans the same way it would look at the life support system or the propulsion system. And it does we do a risk assessment. We lay out all the components of that system that could fail. So, for example, the risk of an astronaut's bones getting so weakened by a one-year transit to Mars that when they wear their 350-pound spacesuit and trip on a rock, they break a hip, which they can never heal, gets quantified. They have an estimate of the chances of that with today's knowledge. And then there's a bar that says what's an acceptable risk for a mission. And so, you know, essentially you do that for every component of the system, the risk of failure of your propulsion system so you never get to Mars, and the risk of failure of your oxygen generation system so that when you get there, you're dead. You know, all of those things get put together, and they try to get the overall mission risk down to an acceptable level, which is generally 1 in 1,000 risk of failure of mission. And so all those pieces are, I think, 78, I may be off on the number, there are 78 human health risks that are quantified in that way. And the minute they can solve the problem enough to get that risk down below the threshold, they move on. And so that's the NASA approach, which means they're basically, they'll give me a hard time for saying it this way, but I think it's the right way to say it, is that the NASA engineering sort of focus and that approach, which is exactly the right way to do space missions, means they're sort of trouncing through the field of daisies of how life works, and they're picking the ones they need and then moving on somewhere else. And so there's just this incredible richness of weird things that people have stumbled across that, that aren't being followed up on because that's not NASA's goal. So the beauty of the designation of ISS as a national laboratory by Congress was that certain key congressional staffers and leaders that have been supporters of the space station for a long time knew exactly that thing. And they said, you know, we need the national lab to open up the ISS to NIH and NSF and commercial organizations like pharma and you know, material science and industry. We need to make sure they have straight line access, that just because NASA's focused on these exploration things, they still need the opportunity to follow up, not 10 years from now, but right now, as those things are going on. And so that's really why we have that structure, so that discovery in general and then applications, terrestrial applications, don't get lost in the engineering march forward to get to Mars.
Can I just follow this? So are you giving astronauts specific medical interventions to lower the risk, at least in the medical side of things? Or are you giving them bisphosphonates from fluorosis?
Right. So we have tested bisphosphonates. Right now, we're not giving astronauts bisphosphonates as a standard medical practice because what we found is if we have them do the right level of high-intensity resistive exercise, eat the right number of calories, which is hard to get astronauts to do because the food doesn't taste that great when there's no convection to make it smell good, and also make sure they have the right amount of vitamin D. If you put those things together, we can actually maintain bone mass density. Now, that doesn't mean the cortical and trabecular structure is quite what you'd want from a fracture risk perspective, but if you can solve that without bisphosphonates, that would be better. Because bisphosphonates are definitely, and those of you in clinical practice know this well, they're really not turning out to be the panacea that we thought they were 5 or 10 years ago. You know, we're seeing problems in patients. There's some problems with GI issues and so forth, which are exacerbated when you're floating in microgravity. So there are a number of reasons maybe not to go the bisphosphonates route if you don't have to. And so every single one of those 78 risks has a story like that behind it, where you're trying to find the optimum solution and solve that and go forward.
We had a question on that.
That's sort of a two-part question for Robert. Basically, you're looking at cranial pressure changes. But in traumatic brain injury, most neurosurgeons and neurologists try to balance both ICP as well as
Wow, well, that's an incredibly difficult question for me to answer, but I guess I can take a stab at it. So there are various things that, and Julie, you might know about this, on how to treat high ICP episodes on Earth using certain drugs and things. I don't know how those would work in space. So I don't know if you have any comment on.
Well, you know, it's a richer story than that because not all astronauts have elevated intracranial pressure, even though all astronauts do have the fluid shifts that show their puffy faces and their skinny chicken legs, we call it. And right, they're perfectly normal. Right, you're not going to be able to manage it that way. Now, there are other tools for managing it. For example, we have a lower body negative pressure device that you can apply and use that to basically suck the fluids down into the lower body. So those kinds of tools would be things that in a crisis situation, even today on the space station, physicians could look at doing. That said, a traumatic brain injury in space, you probably have a lot more problems than just dealing with the swell at that point. So because you're really in field, you're not even in a field hospital situation at that point. You're really in full crisis field medicine. And so you know, one of the things and the risks that folks are dealing with in terms of living in space and carrying people forward in space is even doing surgery is complex because the minute you make a cut, the blood starts going everywhere. So there are a lot of challenges to be faced in dealing with a situation like that go even a lot deeper than managing the pressure. But what I'm most interested about in the ICP issue, and what I really like, is, for one thing, the women aren't having it at all. And so, gee, why not? And some of the men aren't having it at all, but other men are having it a little bit, and some men are having really high levels with high elevated spinal pressures 8 months after returning from the space station. If you do a spinal tap, it's still high in some of them. Others are recovering when they get home. Some, it looks like the swelling of the optic nerve is permanent, and 4 years later, they still have it. So that heterogeneity in response is telling us something about the way that the body responds. And if we could sort out what makes different astronauts different, suddenly you're looking at a genetic window into maybe how you should be treating the patients back on Earth, right? It may be that there are some patients on Earth that you need to manage it more carefully than others, or it may be that opens a window into that some patients would do well with a pharmaceutical intervention and other patients it's not going to help. And so that really ties back to the themes from yesterday about when you do see this heterogeneity, what does that mean for understanding the mechanism? So we're getting ready to fly a PCR machine. We haven't had one in space before, and that will give us that answer, I think. But we just really don't know yet. So the red blood cells hold their shape because that donut shape is regulated by the cellular structure. And Cliff may know more about that. That's kind of your expertise.
You may. No, I think that's more a membrane function than it is a gravity function. So the biconcave disc is maintained, at least the pictures I've seen of the rheology of blood, is that the rheology itself doesn't change with regard to the red blood cell. What does change is macrophages, and the cytoskeleton relationships in macrophages and motility change, and chemotaxis changes as well. But that changes as a consequence of the internal milieu of the cell, but the membrane structure, I think the membrane forces remain the same.
When I was sitting yesterday watching the different forms of metastasis and how groups of cells were working together and how individual cells were sometimes using amine void, all of that stuff would absolutely be affected by spaceflight. So.
Yeah, this is a question for Gwen. Circadian rhythm is controlled by a number of different genes, clock, cycle, and clock hormones. And there are two positive regulatory loops and a negative regulatory loop. And clock hormone is a transcriptional repressor. Does that impact your studies in any way?
Yeah, and actually we've looked at that, and we've looked at the whole circadian complex, of which, at least in mammals, there are about 12 genes. And SRC2 seems to be the controller for BMAL1, which is the second in command. Again, we see difference in BMAL1 enrichment when we knock out SRC2. Now, there are previous experiments that were not coming out of our lab where BMAL1 clock have been knocked out, and they have effects that are independent of the SRC2 knockout. Yeah, we've looked at the circadian complex, all that, and a lot of that's been published by other groups as well. Don't know the answer to that, and it's in fact that's a critical question, Jack. And I will tell you why I'm excited about that question is that we've been discussing that as well. not with regard to space flight, but with regard to mitochondrial energetics. And we have a lovely experimental system available to us, which is heart transplants and left ventricular assist devices, but particularly left ventricular assist devices, where they, in order to put the device in, the left ventricular apex is cored. And we can get that core tissue and have it in the laboratory within minutes. And so we can see what mitochondrial activity was in the evil humors of heart failure, mostly in diabetes, and then change the milieu of the mitochondrial and watch the energetics recover within minutes. But I think that the coactivator effect on mitochondrial function is a really, really critical one. I don't know the answer to it.
The reason I was asking the question, the way it's connected to yesterday, and then whether it's connected causing change in cycle, or whether it's responding to that change. But we heard yesterday that the signature for metastasis due to changes in mitochondria. So
Sure.
at dinner last night, we were talking about what people
Right, with regard to the metastasis, just very briefly, is that the coactivator family, the steroid receptor coactivator family, appears to have a role in deciding whether a cell survives the trip from the primary where it has to pack its own lunch on the trip to becoming a metastasis. And that's just part and parcel of exactly what you just mentioned, which is where's that energy come from? Where's the energy stored? Where's the energy coming from, and what regulates its utilization? Because that would clearly be a target of therapeutic intervention.
This is a down to range question I'd like to ask. So, what concentrations of protein have you used here on Earth, and what are you using in space? And just how you guys grow that protein?
So we make the protein but recombinantly. So we either make it using E. coli, or we use a baculovirus system, which is insect cells, or we use mammalian cells, HEK293 cells. And in those systems, the Huntington protein will have a tag that we can purify the protein with, such as a nickel tag or a CMYC tag. Then for setting up the crystallization trials, we've tried on Earth anything between 1 to 80 mg per mil. We ended up using for microgravity crystallization trials between 5 and 20 mg per mil, depending on exactly which construct and what source the protein was from. So the protein is glycosylated when we produce it in mammalian cells. We sent a few mutants that had regions that did not have glycosylation, and those we produced in E. coli.
So the protein that you're actually sending to the lab, which one are you selecting to make it coli or sensitive?
So we sent 4 different constructs up. So there's a range. So the question is, what happens to the crystals on reentry? And the answer is we don't know. There have definitely been experiments before where crystals have come back unharmed, and there's also been experiments where reentry has been detrimental to the crystals and they've shattered on reentry. And so I think Julie was talking about the possibility of having a way to actually get diffraction from the crystals on the International Space Station. At this point, we don't have anything to really mitigate those effects. And since we've never had large crystals of this protein, we really don't know how it will be affected by reentry. I'm not sure what the plans are.
So the point is that we'll know if they're breaking down on landing. And so if it turns out once we have the X-ray diffraction capability, we'll have a basically kind of a thumb-eye view of how good the quality was. If it looks like once we get them home, that's not it, then we're gonna have to put the smart engineers on figuring out a way. Well, what we would do is we would actually develop a vibration protection system for reentry that would put it under power and actually control that or do something like that. But we haven't put engineers on that problem yet because we don't really know if it's a problem.
Yeah,
Space shuttle experience was that we tended to get things home just fine. Now, the space shuttle was a softer landing, but our Japanese colleagues have been returning things on the Soyuz with the Russians for the last 4 or 5 years, and that's a harder landing than SpaceX, and doing all right. So we're not really sure that it's a problem, but it will be nice to have that insight once we have the capability. And it'll be just nice to know if we're getting crystals, yay or nay, because all of these things that we're flying, and when you're flying crystals, part of the point in space is to grow them very, very slowly, because the lack of convection means that you can get this really beautiful depletion zone around the forming crystal and have just a single molecule cross that zone, and then a really long period of time, and then the next molecule crosses the zone. So when you're working in those kind of slow time frames, if you know that the crystal's forming after a month, then you know that, but maybe you need to leave it up for 2 months. And so the time scale is probably the most unknown part about protein crystallography in space, because it's managing that depletion zone. And since we did a lot of these in the past, all the physics of the depletion zone has been framed out by some European researchers. So it's kind of a new world on the fluid physics side of really looking at that process than it was even 10 years ago with that new data.
there was a question up top. Yes, sir.
So I was thinking about the C. elegans slide you showed, which is paper cells and how they adapt. I know it was in space for quite some time. But when you think about sending nice colonies up, you find certain probably naive to space with a lot of stress. How do you acclimate the mice to this during the space flight using zero gravity? I mean, think about the stress conditions, what effect on the measures you might make in terms of functional responses and challenges. What are the strategies that you use?
Yeah, the question is about mice and their behavioral acclimation to space. And it turns out from studies we've done in the past, what's really stressful is launch, not being in space. So the launch is stressful. It's stressful for humans. It's stressful for mice. There's G-forces there. It's weird. But once that passes, what we see happen behaviorally in the mice is it doesn't take them very long, just like the astronauts, to kind of go, ooh, I can float. And then they figure out how to use their little claws to hold on to the sides of the cage. And so then all of a sudden, they've renormalized their environment. In the same way mice in a barn are going to be crawling up the walls and across the ceiling, they're acting the same way. So the stress responses tend to go down pretty quickly.
Yeah, we actually have video evidence of that acclimation, which would defend that. And what's interesting in this new environment, most, if not all, of the rodent experiments that have taken place were during the period of space shuttle, where there was at maximum a 14-day sortie. At least now with the International Space Station, and it obviously is dependent on the research objective, but you have a much longer time in which the rodents could be exposed to that environment, and so you can probably better defend acclimation.
The other problem that occurs, and I only know this anecdotally, is that the male mice retain their charming personalities. And so that at this point, the problem is keeping the mice, well, I think they're only flying female mice now, right?
Right now, yeah, but there will be male mice
and
flying.
it's because of the delightful interactions that the male mice engender. But when you want to start to do reproduction experiments, and particularly regarding embryogenesis, that's going to be a real challenge as to how to manage the sociocultural acclimatization of the mice.
We are scheduled to take a break, so I want to thank each of you once again for taking the time to present today and to answer some of these questions. Very good presentations and very good discussion, so thank you very much.